Well, thank you so much. It's really a pleasure to be here. And yeah, please ask questions in the middle. It will be much more interesting. So we are mainly going to talk about work that we've done in collaboration, well, obviously within our lab with Kelly and Joe and Debbie, but also in collaboration with the Oxford Machine Learning Group, in particular Pascal, which was a PhD student that we worked really closely while working on what we're going to tell you about. So the question, the problem we're going to be discussing today is probably one that most of you are familiar with. While sequencing is becoming more and more ubiquitous and available, there's this hope of using it in the clinic for diagnostic and preventive care. But the reality is that what we know about the clinical implications of genetic variant is very much limited, and it's in infancy. Just to give you a notion of the size of the problem, even if we just focus on the problem of missense variants, if we look at NOMAD, there's about 4.8 million missense variants seen in the people in NOMAD, but less than 2% of those have any type of clinical interpretation in ClinVar. And this knowledge is not only extremely sparse, but it's also very biased, both in population, because some ethnic backgrounds are much more sequenced than others, but also in terms of genes, because what we know about some genes is, we know things about genes much more than others. So sequencing has been helping with this whole process of variant annotation. We now have the possibility of doing familial and population studies at a much bigger scale than we were able to do before. And also there's now the development of high throughput functional assays more and more coming up, specifically developed to answer and tackle this question. But the size of the problem is such that to really answer the problem of variant annotation, we're going to have lots of orthogonal sources of evidence that are scalable and that can be combined with each other. So what we're going to ask today is if generative models of sequence data across diverse organisms can help this task. And in a sense, looking at conservation throughout evolution has been always one of these tools used for variant annotation. You might be familiar with tools such as polyphen and SIFT that look at column conservation to predict the effect of genetic variation. But recently, there's been a huge amount of progress in developing deep learning models specifically to biological sequence data. And these models have had a big impact, or are having a big impact, in protein design, forecasting pathogen evolution, protein RNA structure. Probably you're very familiar with protein structure in this domain. And I suppose that, okay, and here I'm just pointing out some work that our lab has done in this space, but really this is kind of a big effort from the community in general. So what we would like to ask is if this progress can also translate to the exact problem that we want to answer today, that is a problem of clinical interpretation of genetic variants. So what we mean by looking at sequences across diverse organisms is actually super intuitive. When we look at sequences across very diverse organisms, we're looking at millions of years of evolutionary experiments that have been basically selecting for fitness. So if we look at the distribution of the sequences, and if we model the distribution of the sequences, we can hope to capture the patterns that encode these fitness constraints. And this is exactly what we mean by a generative model. It's a model that describes the distribution of the sequences. So here I wrote P of X, where X is the sequences and theta is just the parameters of the model. So once we have such generative model, once we have the model of the distribution of the sequences, then we can query if a new sequence we haven't seen before, say from a patient with a mutation that we want to know the effect of, what is the probability of that sequence according to our model? And the idea is if the sequence is less probable, it's probably less fit. And therefore we have a notion of fitness, and this is kind of like this log odds ratio will come up, you will see throughout the talk very often. That is basically just the probability of the variant or mutant sequence over the probability of the reference sequence according to our reference model.
So yeah, so just to explore this idea in more detail, we can go back to this idea of column conservation. So as Mafalda says, this this model has a long history, and intuitively, if we look at a multiple sequence alignment of sequences from diverse organisms, and we see that an amino acid at a particular position is conserved across diverse species, then we would expect that changing that amino acid for another one that has very different properties is likely to disrupt the function of the protein. So what we want to do is take this sort of intuition and recast it as a probabilistic statement. And it's a very simple model, so there's many ways one could do that. But for the purposes of this talk, it's helpful to think about this in terms of a Boltzmann distribution, where the energy function is just a linear sum of terms. And the notation here is that xij represents indicates the presence of amino acid J at position I. And these coefficients, Hij, basically are a measure of conservation. So when we write the model in this way, we can then sort of have a quantitative measure of conservation through this log odds ratio that Mafalda just described. But what's nice about writing the model this way is that it immediately suggests ways to extend the model. So the first thing that might come to mind is going beyond linear order and thinking about including quadratic terms. And this brings us to a model that has many different names. Mafalda and I, we come from theoretical physics. So for us, this is the Potts model. And the key addition here that you get with these pairwise terms is basically you account for covariation between amino acids at distinct positions in the sequence. And this model actually has a really long history in our lab, not for mutation effect prediction, but for structure prediction. And the reason for this is that if you take the magnitude of these Jij terms, say like computing the Frobenius norm, for instance, then you can plot the sort of the largest Jij terms as a function of position, and you get a map like this. And what's lovely is that if you compare this to the crystal structures that people obtain, then you often get a really fantastic overlap. So this and these contact maps are kind of like a crucial part of structure prediction. So these are important for things like AlphaFold, if you've heard of that. And indeed, our lab has a long history in using this for structure prediction and looking at interactions more generally, actually. However, for the purposes of the talk today, we don't care so much about structure prediction. We just care about this. Okay, so yeah, so what we can do is compute all possible single amino acid substitutions, for example, and we can represent these as a heat map like this. And what the majority of these experiments, at least so far, do is also try to do the same thing. So try to do an exhaustive study of all possible single amino acid substitutions. There are some really interesting examples that go beyond just single substitutions, so looking at doubles or selected sets of high order, but the majority so far focus on single amino acid substitutions. So what we can do then as a way of benchmarking our model is compare the predictions from our model to the predictions from the experiment. And what we see is that in many cases, we have a really fantastic Spearman correlation with these experiments. So to use these as a way of benchmarking the models, we don't look at one deep mutational scan, we collect as many as we can. And here are some examples from the Rieselman and al. Paper. And actually, we're hoping to release an update of this soon with many more. But the basic idea is that we can compute the Spearman correlation for each of these experiments. And in doing so, we now can compare our model's performance across a wide range of experiments. And what you see is what the maximum possible Spearman between our models and the experiment could be. Because, for instance, it's an upper bound just on the correlation between replicates, for instance. So why you can't look at can't take too much meaning from the overall value of the Spearman, we certainly can use it as a way of comparing one model against another. So if moving from site-independent model to a pairwise model gave us some boost in performance, how could we push this idea even further? So the first thing that might come to mind would be to go from quadratic order to cubic order. As far as I know, this hasn't been fruitful for anyone. It certainly hasn't been fruitful for us in the lab. So in recent years, we've moved in a slightly different direction, which is towards sort of deep learning methods. And there's been a sort of flurry of activity in this space, particularly in the context of protein design. And there's many, many different models out there now. The one we're going to focus on today, though, is a variational autoencoder. So the basic idea with a variational autoencoder is that, well, sequences are these incredibly high dimensional objects. And so almost no matter how large our multiple sequence alignment, we will only see a vanishingly small fraction of the possible state space. So this, so in that sense, the data is incredibly sparse. So one way to get around this is to postulate that the data that we observe can be explained as being generated by some much smaller set of variables. So some lower dimensional space, which can represent the data equally well. So for this, we imagine that there's 50 dimensional latent space. And we sample these random variables from some simple distribution. And then these variables are passed through some non-linear mapping F to become the probabilities from which we then sample the sequence we observed. So you have a categorical distribution at each site. So to get some intuition for this, we can think about different limiting cases. So in particular, if F is independent of Z, or if we condition on Z, then the model just reduces to a site-independent categorical distribution at each site. So this is precisely the site-independent model that we started with. So one way to think about this model is sort of as an infinite mixture of site-independent models. Another way to think about this model, or another interesting limiting case, is when F is linear up to a normalization, in which case this is just principal component analysis. It's probabilistic PCA. So another way to think about this model is as a generalization of principal component analysis, where we now allow F to be nonlinear, and in particular, we parameterize it with some large three-layer fully connected neural network, which, as you may be aware, is a very general function approximator. So the idea is that we now allow F to be some very broad class of functions. So just to expand on this idea of principal component, or generalizing principal component analysis a little bit more, we can look at the latent representation of the training data. So what we've plotted here is the first two principal components of this latent space, where each point now represents a sequence in the training set. And we've colored them according to their Hamming distance from a reference sequence. So as a human sequence for the tau protein. And what I find pretty amazing, actually, is that if you plot all possible single amino acid substitutions, they're all in this tiny pink region here. That's the exhaustive set of single amino acid substitutions. So I find it kind of incredible that, the model is training on this incredibly diverse set of sequences, and yet we're trying to benchmark the model or make predictions for variations in fitness on the scale of this dot here. Yeah, I think it's really remarkable that these models can make any use of these distance sequences at all, actually. Nevertheless, it seems to work remarkably well. So when we compute the Spearman correlations against experiment with this model, again, we see a nice boost in performance relative to the POTS model.
Yeah, right. So because the VAE is performing, as we see it, state of art for this task, we decided to use it for the particular problem of clinical interpretation of variants. So what we did is that we took a VAE and we optimized it for this particular problem. In particular, we made it as robust across protein families as possible and quite scalable. And what we get out of this is this log odds ratio that we talked about before, but we called it the Evolutionary Index in the context of our work. So like we discussed earlier, this has kind of like an intuition for the fitness of the variant, but it's quite hard to map it to a clinical interpretation, so a scale of pathogenicity of the variant. So to do this, what we developed was, on top of the VAE, we built a Gaussian mixture model, and I'll explain it in a second. But what we get out of it is a pathogenicity score that basically just goes from 0 to 1, with 1 being more pathogenic for each variant, and also a class assignment where we assign each variant to a cluster of benign or pathogenic. So let me explain a little bit more detail how we do this Gaussian mixture model step. So when we plotted the distribution of evolutionary indices for every protein, we realized that the distribution was my model for some of these proteins. And this kind of like motivated us to basically just cluster the distribution of the evolutionary indices in 2 clusters. And we postulated that one was corresponding to benign variants and another one to pathogenic variants. And this sort of like super naive approach actually paid off quite well. In practice, what we do is that we have a mixture of Gaussian mixture models where we have a protein-specific GMM and also a global GMM, where we take the evolutionary indices of every single protein that we studied. And we studied about 3,200 so far now. And you can see the distribution is not by model at all when we look at all the proteins together, but still we just do a mixture model of two Gaussian clusters, and the results work pretty well. We call this model EVE, Evolutionary Model of Variant Effects. And we studied, like I said, about 3,200 proteins so far that are known to be involved in disease. To basically check the performance of this model, we computed AUCs, so area under the ROC curve, on known annotations from CleanVar. And this is basically the distribution that we get. Overall, we have an AUC of an average AUC of 0.91. What you can see here is that a lot of the proteins that we were studying have actually very small number of known annotations. So it's quite noisy to compute AUCs, and you can get like noise either up or down on the AUCs. So we also checked what was the distribution of the AUCs only for the subset of genes that have at least 10 known annotations for computing AUCs, and indeed we see that the performance goes up slightly. So like I was saying, another thing that we can do is to actually do a classifier where we classify variants as benign or pathogenic. And because we do this with a Gaussian mixture model, it's a probabilistic process by heart, so we have a notion of uncertainty. So what we can do is to have an interplay between how many variants we want to classify versus how much accuracy we want to get of our classifier. So for example, if we classify, or if we give a class assignment to every single variant in our 3,200 proteins that we studied, we basically have assignments for 36 million variants and an accuracy of about 85%. But if we take the most uncertain assignments as uncertain, and what we get is that we can increase our accuracy. In particular, if we set the 25% most uncertain assignment as uncertain and just give assignments to the remaining 75% of variants, we have assignments to 27 million variants and an accuracy of 90%. Throughout this talk, we are going to show results using this threshold, the accuracy at 90% threshold. But in reality, this is not how we envision people using this uncertainty as a measure, as a tool for better predictions for the model. What we think is that the most appropriate way to use this is in a gene by gene basis, because different genes will be associated with different diseases, with different severities or different treatments. So the requirement for precision recall is different in a protein by protein basis. So we imagine that when people are interested in specific genes, they will use this uncertainty in different ways. Nevertheless, just to give you an idea, a global idea of what this means for variant annotation, this is what was currently known. So basically, these are variants of unknown significance that we just used. All variants that have been seen in NOMAD and UK Biobank that have no known annotation in CleanVar, and these are the annotations currently in CleanVar for these variants. And if you take these if predictions with a 90% accuracy, you can see that we increase the amount of predictions quite a lot.
So we also benchmarked the method against other popular variant effect predictors, and for this we used two different benchmark sets. So the first one, the x-axis here, is the clinical labels that Mafalda was just describing. So the ability to separate benign from pathogenic variants. And and I should explain the notation here. So if the dot is filled in, this corresponds to an unsupervised method. So a method like ours that is potentially trained on just evolutionary data or something like that. And if it's an open circle, then this corresponds to a supervised method. So a method that saw clinical labels during training. And there's a bit of a problem with this benchmark, which is that there's a circularity here. So many of these methods do not make their test training sets publicly available. Also, the intersecting sets of them is complicated too. So we just simplify things by testing the methods on the full set of clinically available labels. But the downside of this, of course, is that we're using for testing labels that were used in training these models. So there's some circularity here. Nevertheless, it was a really nice surprise to us to see that our model outperforms on this benchmark, despite the circularity. And yeah, this is not something we were expecting going into this project. We thought it was just a worthwhile contribution to have an unsupervised method anyway, because of how these methods get used in variant annotation. But yeah, it turned out that actually evolutionary data seems to be a more compelling source of evidence than we were expecting. So yeah, it seems to be able to stand alone. Nevertheless, to try and have a second sort of less circular benchmark, we also looked at concordance with deep mutational scans, as we were describing earlier. And here we restricted to deep mutational scans that were developed specifically to do the same task, so specifically for variant annotation, trying to predict benign versus pathogenic labels. And I think that's about 14 deep mutational scans that we have in this set. I think that there's more all the time, but that corresponds to still hundreds of thousands of independent experimental measurements. So although it's a smaller set of proteins, it is a less circular benchmark. So it sort of complements this other one. And so it was very nice to see that we can achieve state of the art on both of these benchmarks.
Right. So far we've only talked about using deep mutational scans almost as a ground truth that we check if our model is comparable with these experiments. But the reality is that these experiments are developed for exactly the same task. So we can now ask, how does our performance of the model at predicting clinical annotations compares to the performance of experiments at predicting clinical annotations? And here is an example of this comparison for P53. So these are the experiment predictions from Giacomelli et al.'s paper. And here in the x-axis is just a position along the protein. So these are experiments and these are model predictions. All dots here are clean var labels, benign and pathogenic, and all the crosses are variants of unknown significance that have been seen in the population. And here, red crosses and blue crosses are just our predictions from our model at these 90% accuracy thresholds. So what you can see here that the AUC for our model is slightly higher than the AUC for the experiment, and we compare against different experiments who basically have the same kind of trend. But overall, I think that the most important message is that we compare on par with experiments. And I think that this is something that we're really excited about, because it basically says that evolutionary evidence, evolutionary information can be used in a very similar way to high throughput functional assays is used currently for variant annotation. And these are very complementary, independent sources of evidence, and therefore they can just be used together in concordance and build on each other. And I think that this is something that is really exciting and something that's, really was something very important for us. Okay, so because it's almost noon, I'll try to go through these slides quite quickly. So this is an example of the output for MSH2. So MSH2 is in a complex with MSH6 in a DNA mismatch repair protein complex, and it's involved in lots of hereditary cancers. And when we plot our scores in the 3D structure, we can try to identify these pockets of high pathogenicity. And not totally surprisingly, the pockets of high pathogenicity are, locations of ligand binding and DNA binding, etc. What was surprising for us, so here, for example, this is the place where the ADP binds to the complex. What was surprising for us is that some of these locations in these sites that are actually some of the highest high pathogenicity in our model have been seen in the population, and they weren't annotated. So it really just tells you how much of a problem we still have now, and how much models, and the experiments can help, and how much further we can go with variant annotation. Okay, so this is the other side. This is MSH6, and you can see that there's also a lot of pathogenic variants at the DNA binding site. And this is basically an equivalent exercise, but with the ion channel complex. This is SCN1B and the heat map for the scores of SCN1B that binds to SCN4A, again, involved in lots of disease, in this case, cardiac and neurological disease. And again, Most of the pathogenic variants are either in locations where SCN1B is in contact with SCN4A, or also on like the IV's hydrophobic core of SCN1B. And here, you can see that there's a lot of pathogenicity at the ion core of SCN4A.
So yeah, as we mentioned before, kind of a key property of the model, or model, something we think that makes the model very useful in practice, is that it's unsupervised and that it's trained on just a single source of evidence, which is this, sequences from diverse organisms. The reason this should be helpful is because it makes it easy to incorporate into variant annotation pipelines, which seek to combine different orthogonal sources of evidence. So it can really be just treated as a single source of evidence. So this plot basically just continues the Sankey diagram that Mafalda described earlier. So on the left, we have sort of the known annotations in ClinVar. In the middle, we have the predictions from the model using, again, just as an example, this 90% accuracy cutoff. And then on the right-hand side, we have the annotations that we found additional evidence for. So, things like, is the variant seen frequently in the population, or are there other pathogenic variants known at the same position in the protein sequence, things like this. And basically, when we try to combine our model with other sources of evidence, we find supporting evidence for about the reclassification of about 256,000 variants of uncertain significance. So this estimate, this gives you some sort of estimate of the potential impact, but it is admittedly a bit of a crude estimate. Really, how we envisage the method being used is being treated much more like how experimental assays are treated. So, each experiment is validated on a case-by-case basis, and I think that the appropriate way to use these models would be the same. So, for each protein, validate the model independently. If there's many labels available for validation in that protein and it performs well, then you can reach a conclusion that the model might be a very strong source of evidence. However, if the model performs less well, or if it seems to perform well, but there's less labels for validation, then you can't be as confident about the predictions, and therefore it probably should only, should be used as a weaker form of evidence. And indeed, this is what some people are doing now. So in particular, a company Invitae has incorporated our model into their pipeline, and they're doing something like this. So the strength of evidence that the model provides varies on a gene-by-gene basis and on a cutoff dependent basis. So it's a much more refined approach, and they're basically doing their own independent analysis of the model now. And I think there's going to be some report in the first quarter of this year. So looking forward to seeing what their results are.
Yeah. So just to finish off the discussion about this model, we have all our results available in our web server. You can download the results for individual proteins or for everything. And we are trying to, I can show you, this is the kind of stuff that you can get out of it. And we are currently have models for about 3,200 proteins, but we are hoping to scale this up to the whole human proteome. If there's anything that you are particularly interested in, please just email us and we'll just try to speed things up. Right now, we already have 600 requests for other proteins, so we're trying to deal with this queuing and using resources, computational resources in our cluster for this. But yeah, so the idea is to basically just scale things up for the whole proteome. Okay,
so this basically brings us to the end, but we just wanted to finish with a couple of comments on kind of future directions. So in this work, we've kind of painted the picture that moving to sort of more expressive, bigger models has been fruitful, and it has been somewhat fruitful. So, moving from the site-independent model to the POTS model certainly improved performance, and moving to the variational encoder also improved the performance. So there's a natural question of can we just move to bigger models and bigger data sets and get better performance still? And in recent years, there's been definitely a surge of interest in moving to alignment-free models for many reasons. There's a number of limitations with using an alignment-based method. So these are extremely appealing methods for a number of reasons. But a sort of subclass of them that seem very promising in some ways are these extremely large language models, so transformers and things like this. And while they have many appealing properties, their performance gain in terms of unsupervised variant effect prediction has been somewhat underwhelming so far, at least. So I think so far the variational autoencoder continues to still be state of the art in terms of unsupervised variant effect prediction. And so the question, I think one of the questions is why this is, and recently some work led by Eli and Alan in our lab seems to be shedding some light on this. So I'm not going to go into this in any detail, but they basically prove that, well, something we know about protein sequence data is that it's not statistically independent and identically distributed. It has a phylogenetic structure. And this is something that most models are neglecting. And this looks to be a bit of, well, quite a serious problem in some ways. So what they prove is that fitness and phylogeny are non-identifiable. So you can describe the observed data equally well by exchanging fitness and phylogeny aspects of your model if you have a joint fitness and phylogeny model. And to gain some sort of intuition for this, you can think about a situation where you have two sequential neutral amino acid substitutions. So imagine we start with GLAKP, and then we get a substitution from A to G, and then a statistically independent event occurs, which is that K gets substituted to E. Now, a good density estimator of the data distribution would conclude that that GLAEP, well, a good density estimator would score this very low because it's never been seen in the distribution. However, if the underlying distribution before phylogeny was statistically independent, then GLAEP would be a perfectly fit distribution. So in this situation, a site-wise independent model would make a better inference of the underlying fitness than a superior estimation of the density distribution. And so yeah, basically, I think the takeaway from this is that while going to bigger models is almost certainly going to be beneficial in many ways, I think that we are working on transformers as well. There's no guarantee that moving to bigger models or simply just going after better density estimation is going to be fruitful by itself. It seems like we need to think very carefully about how to combine these sorts of methods with sort of biological knowledge. And I think that's partly why using multiple sequence alignments has been fruitful, because there's decades of biological knowledge baked into those alignments. And yeah, I think this paints a very exciting situation for model building going forwards, because it's really about trying to incorporate biological knowledge into the model building process, which at least to us as physicists is a very appealing thought. Okay, so that really does bring us to the end. So just to summarize, variant interpretation is a megalomanous task. There's a huge number of variants of uncertain significance, even if we think about missense variants. And that's something that's holding back the usage of sequencing data in the clinic. So to overcome this problem, we're going to need, it's a global effort. It's going to need more population level sequencing, much more high throughput experimentation. And the goal of this work really was to try and bring or revisit another source of evidence, which is the usage of sequences from diverse organisms. And we felt the time was right to revisit this because of the recent progress that's being made in sort of deep unsupervised modeling of sequence data. And yeah, it turns out that this data, and combined with these models, seems to provide really quite a compelling evidence for variant interpretation. So I think, well, we hope we've convinced you that it's sort of an appropriate time to reconsider how this data is used in variant annotation. Nevertheless, I think it's absolutely clear that we have barely scratched the surface of what these models can do. I mean, we clearly, there's lots of things we don't understand about them still. And I think there's lots more that we can learn from building better models of protein sequence data. Yeah, so that brings us to the end. And I finally just want to thank our lab. So yeah, our lab is growing. This is kind of the members from the last few years that have all been very involved in method development in one way or another, although everybody has some different applications ranging from protein design to pathogen forecasting. But I think we all work very closely, especially when it comes to the method development side of things. So I think this work has benefited from discussions with everybody. And in particular, I would like to thank Debbie, our mentor, and also she was very hands-on in this work. So she was not only a great mentor, but a very close and great collaborator in this work as well. So yeah, thanks to her too.
Thank you.
That brings us to the end. Thanks.
Awesome.